Hi Bart, nice to speak with you again.
Same, thank you for having me, Narina.
What are you mostly busy with right now?
I guess I'm working with few organizations on synthetic biology and gene drives, specifically for nature conservation or biodiversity conservation. So that includes IUCN, which is International Union for Conservation of Nature, but also UNEP, which is United Nations Environmental Program. For them, I'm actually preparing a chapter for their report, annual report. And yeah, basically, I try to kind of raise awareness of the issues related to synthetic biology and gene drives.
Why synthetic biology?
Synthetic biology is basically this new and kind of emerging field that actually emerged in a way from biotechnology. So you can think of it as extension to biotechnology, but it's actually more sophisticated and in a way more powerful. So synthetic biology actually allows you to change genetic code and basically develop some synthetic organisms or biological components or biological parts. And synthetic biology actually uses similar techniques to biotechnology, but many of these techniques are actually more sophisticated.
It is sophisticated, but there are interesting developments outside academia going on, right?
Yes, so because synthetic biology is quite exciting, and you can also think about synthetic biology as hacking biological code, because in a way, DNA is biological code that can be hacked potentially. So I guess hacking kind of moved or expanded from computer kind of related hacking to recently to biohacking. There is this rapidly growing movement of open science movement of so-called citizen scientists and biohackers and different do-it-yourself biology enthusiasts. So the movement is actually called do-it-yourself biology or DIY biology. And basically it's a movement of hobbyists that normally don't have prior experience in this field, but they are trained by experienced scientists from pharmaceutical companies, from academia, and so on. Many actually proper scientists are also involved in this open science movement. And basically these scientists and citizen scientists try to basically develop different applications, different novel applications for synthetic biology by altering existing biological systems. And this is done mostly in the garage or in the kitchen most of the time. But there are also specialized labs across the globe that are basically rapidly growing in numbers that provide specialized facilities and equipment to basically attract these citizen scientists to provide them tools to basically do their activities or experiments. There was an article back in 2004 where author claimed that was back in 2004, so the equipment was much less advanced and it was much more expensive. But even then, he claimed that garage labs kind of era is upon us, and you can basically build a garage lab for as little as $1,000 by sourcing equipment from eBay. So that was quite exciting. This article was published in Wired magazine, which is quite prestigious, and because of that, it got a lot of publicity, and many people actually started following that. Another thing that basically kind of skyrocketed this international and quite exciting movement, but also quite scary in a way, was so-called iGEM competition. iGEM stands for International Genetic Engineering Machines. So it's basically a student competition. Initially, it was student competition aimed at undergraduate students, where they basically design different synthetic biology experiments and they try to develop as part of this competition. I believe it was in 2003 when one of the teams in the US, student team, decided to reproduce gene drive described in one scientific paper. This team actually failed, but everyone basically kind of freaked out, everyone in the field, so academics and also people that are governing the whole competition. They in a way freaked out because so far gene drives are still done only in the lab on small populations, very controlled environment. no one has ever released a gene drive to the environment because no one really knows what will happen. And because this technology is so powerful and so hard to control, it will allow you to alter entire populations. Everyone is kind of scared of doing that. But this team of students actually tried to reproduce a gene drive. They failed, but afterwards the team leader actually said that because it attracted so much controversy, it actually made him work even harder to make this happen, which is kind of you would think like, oh, what a guy, you know? Everyone is telling you don't do it, but he's like, oh, all this controversy actually made me do it even more.
Could you tell me more about gene drives?
So gene drives is this technology which allows you to basically modify genes in living organisms so that the desired gene will have close to 100% or 100% chance of being inherited by the offspring. Normally, most of the genes actually have about 50% chance to be inherited, but gene drives, they have actually close to 100% chance to be inherited by the offspring. And here I'm talking about artificial gene drives, but there are also natural gene drives. So natural gene drives are basically very similar kind of forms or genes that basically have much higher chance of being inherited by the offspring. But we humans recently actually developed ways to create these artificial gene drives, and as such we can use this technology or a tool modify entire populations. This is kind of exciting, but it's also quite scary, and it's a very powerful tool.
How could we use gene drives? What kind of applications could we use?
Gene drives are quite exciting for a number of reasons, because there is a lot of discussion regarding gene drives and their use in eradicating malaria. So you can basically alter mosquitoes that vector malaria. You can switch off the gene that actually allows them to vector malaria virus, so that they are unable to basically transport this malaria. And by doing that, you will be able to eradicate malaria pretty much completely even. And this is massive progress, because Africa and many other parts across the globe, many other areas and countries across the globe are still suffering from malaria, and this is a really big problem even these days. There is also a lot of discussion about using gene drives, for example, for making coral reefs more robust to environmental conditions. So you basically, in this case, in this application, you would be able to alter genes of the coral reef symbionts so that they are less prone to acidification of the oceans and also growing water temperature. And by doing so, you effectively make them more robust to these environmental conditions. And many believe that this is a way to actually preserve coral reefs across the globe. There is also a lot of discussion about gene drives being used to eradicate invasive species. Even in Australia, the government announced that they are looking at gene drives to eradicate feral cats. So these are cats that escaped from the homes and they, in a way, became feral, so wild. And these cats, according to a study done in Australia, they kill over 1 million birds every day, native birds in Australia. So this is a large number, but also, in a way, I don't like the idea of using gene drives to basically eradicate these cats. Like, you know, using this new technology, very promising technology, for kind of genocidal applications. It's, in my opinion, it's a bit unethical. There are many ethical concerns about using gene drives for this type of applications. So yeah, I guess everything comes down to ethical issues regarding both synthetic biology and gene drives and other related tools, and also policy, so regulating the field, governing it, and so on.
Why do we have to speak now more seriously about regulations for synthetic biology and gene drives?
Okay, so gene drives have been discovered I guess, few decades ago. But these were natural gene drives. So these are naturally occurring genes that have a higher chance of being inherited by the offspring than regular 50%. But I guess with technological development in biotechnology and synthetic biology, recently we came to a point where we developed new genetic engineering tools, including CRISPR, which is basically something that we kind of biomimicked from viruses. So we kind of copied cells' immune kind of behavior to work as a tool for genetic engineering. And with this CRISPR-Cas9 tool, it becomes much faster, much easier, and much cheaper to basically precisely alter some sequences of DNA. And because of that, gene drives became real. So it wasn't really possible, or it was possible actually before development of CRISPR tool, but it was much harder to do so. It was less precise, it was much more expensive, and it required much more sophisticated skills. With these new tools, it's much easier, faster, and cheaper. So gene drives are real. There are already many applications of gene drive being tested in the lab. No one kind of dares, I guess, no real scientist dares to basically release them to the environment.
What are the main challenges, in your opinion?
So there are many questions related to gene drives, like reversibility. What do you do if something goes wrong? So there are researchers trying to actually design so-called daisy gene drives that will only work for a number of generations and then they will switch off. They also try to find kind of safety switches. In case if something goes wrong with the gene drive, they actually try to incorporate a safety switch that will allow to switch off this gene drive if gene drive have kind of harmful effect on the host. So yeah, there are many policy issues, but also many kind of environmental and health issues related to gene drives and synthetic biology in general. Many tools that have been developed for these two technologies are quite new. And because of that, governments are not really good at keeping up with all these technological advances. And there are barely any regulations, but I guess because of that, the field is quite exciting, especially for someone who likes policy or policy making. There are many promises that these technologies can bring, but also many challenges, like how do you make sure that unexperienced users or users that are not aware of ethical issues or biosafety issues are basically doing it right. So it's not only do it yourself, but also do it right. Both fields, synthetic biology and the subfield of it, which is gene drives, requires some strict regulations and governance to basically make sure that they won't cause harm, they won't basically affect environment, biodiversity, and human health. But at the same time, these regulations should allow the continuous progress of the field because these technologies can bring a lot of good. Eradicating malaria or making coral reefs more robust to climate change are only two applications, but there are basically uncounted number of applications.
In the ethical debate over synthetic biology, the formula playing God is widely used. Are we playing God?
I guess we are in a way, and gene drives, as I said, they are still only applied on a lab scale in very controlled environment. No one dares to release them to the environment just yet because no one can actually foresee what the side effects might be. And because of that, many of the scientists that don't want to be blamed for releasing this potentially harmful technology to the environment. Last time we talked about nanotechnology. Some of the challenges that we are currently facing from nanotechnology, we would never think about them in the past that they can happen, but they did. So there will be also many challenges that we cannot even imagine currently. We need to kind of look into the future and kind of identify what potential challenges there might be and try to kind of address them via policy, I guess. But as you said, these two fields are often referred to as artificial life or man-made life, which is basically kind of a relation to life made by God. But instead of being made by God, it's actually man-made.
What are the main points that you are going to present in the policy papers?
Some of the major points are, I guess, long-term studies have to be incorporated in every new synthetic biology and gene drive development. We need to make sure that there are no side effects, and actually to establish that it requires long-term studies. Also kind of incorporating these safety switches in these developments, because in case if something goes wrong, we would like to have a control and kind of be able to switch it off. Also application of precautionary practice, basically that says that if the technology can potentially pose harm, you should kind of avoid using it. One of the paradox of the gene drive technology and synthetic biology is that when you try to do risk assessment, actually doing risk assessment can pose risk itself, because to do risk assessment, you actually need to apply it in certain case study. And by doing so, you effectively kind of pose risk to whatever you do. So it's kind of a paradox to assess the risk. You actually can pose risk. But basically things like that. So together with Global Young Academy, we are developing currently a policy paper. It was supposed to be published as a policy paper for G20 this year, but I guess we will do it next year with G20 or some other international organization. But we are developing that, so hopefully GYA can pioneer this field by introducing this policy paper. Also, in terms of do-it-yourself biology, I guess strict governance is required, and also I guess there has to be a collaboration between these citizen scientists and real scientists. At least some communication, some skill transfer to basically make them more aware of what they do and what are potential threats of their actions.
Nature worked for billions of years to make us and the other species. But now we could make some changes, or we could make changes pretty quickly.
Yeah, I know this is quite crazy, but it's true. Biology took many years to basically get to a point where we are currently. The evolution is quite a slow process, and we are trying to basically make it faster. But it's not necessarily good. Like, we will try, maybe it will bring good things, maybe it will bring bad things, but let's try, you know. If we don't try, we never know. But also, on the other hand, because evolution is so slow, and the changes that we are currently doing to the environment and causing climate change are so pronounced, that maybe actually we need this technology, and maybe actually we need to kind of make evolution faster in order to survive.
What are your next steps? What are you looking forward to, Bart?
So the next step, I guess, is to finish these two reports and advise on policy steps. But also Global Young Academy, which I'm part of, is taking part in CILAC Forum, which is a scientific forum for Latin America. And this year it will happen in October in Panama City, in Panama. Global Young Academy was accepted to host a panel discussion on do-it-yourself biology and basically different entrepreneurial opportunities that this growing movement of citizen scientists can bring to Latin America. We'll also, of course, talk a bit about challenges, but I guess we don't want to scare our audience too much, so it will be more about opportunities this time. but challenges, of course, will be mentioned. So policy challenges, governance, and so on.
Thank you so much, Bart, for this conversation, and speak to you soon again. Bye.
Thank you. I will try to make sure that our panel, GYA panel, will connect with you from Panama. Thank you for having me.